Hi, and I would like to thank the organizers for allowing me to give this talk. I'll be talking on misfolding of acid sphingomyelinase associated with Niemann PKB as a risk factor for the development of Parkinson's disease. So I'll start with a general introduction for this audience, and whoever is familiar with it, I'm sorry for the introduction. So as we know, all lysosomal enzymes are part of the secretory pathway, which means all the proteins are synthesized on ER-bound polyribosomes, and I don't know why you don't see. The proteins are synthesized. You don't see the yeah. Okay. But if I don't, okay. So the the proteins, the lysosomal enzymes, lysosomal proteins are synthesized on ER-bound polyribosomes. They enter the ER, and if normal proteins, if the proteins are normal, immediately upon entrance into the ER, they undergo glycosylation and folding. Once they are correctly folded, they will live in a vesicle. In a vesicle, and if we are talking about lysosomal enzymes, they will reach the lysosome. Even among normal proteins, of course, we have some cases of misfolding or unfolding, and in that case, the molecules will undergo folding attempts. If they are not successful, the misfolded or unfolded protein will leave the ER outside the cytoplasm, will undergo ubiquitination and proteasomal degradation in the process known as the ER-associated degradation. What happens when we have mutant proteins? In a lot of cases, not all of cases it turned out, the more molecules are immediately recognized as misfolded, and therefore a lot of the mutant molecules are retained in the ER for refolding attempts. If they are successful, some mutant molecules, of course, will leave the ER toward the lysosome, because we do have mutant molecules in the lysosome, as we know, with some activity in some cases. But most molecules, or a lot of molecules, will find their way to the proteasome to undergo ER-associated degradation. And so we have less steady-state protein in the cell. And also, since a lot of the molecules, mutant molecules, undergo ERAD, only a little or a smaller part of these molecules will reach the lysosome, and therefore we have less activity in the lysosome, not to talk about the fact that the mutant molecules themselves don't have normal activity because they are mutant. We can do it from here. Okay, so this retention of molecules in the ER for refolding attempts causes stress in the ER. And this stress activates the stress response known as the unfolded protein response, which is a machinery based on 3 receptors on the ER membrane. And these receptors don't work alone, cannot work because they are associated with BIP, and BIP holds them inactive. Once there is misfolded protein in the ER, BIP dissociates from the 3 receptors and become involved in the folding attempts. These receptors are now free to undergo processing and activation, which ends with transcription factors that enter the nucleus and turn on UPR-related genes. Okay, and the three receptors, one, we have three arms, the IRE arm, PERK, ATF6, doing different things, and we test unfolded protein response by turning on of the UPR related genes. One of them is PIP, another one is XPP, splicing of XPP. So I'll show RNA levels going up due to the activation of the UPR. So in the past, we have shown that that UPR exists in two diseases. One is the disease I'm working on for so many years, and we have shown that misfolding is a major determinant in development of Parkinson's disease in a fly model. So we show that once we express a misfolded protein in the dopaminergic cells of the fly, Parkinsonian signs develop. Okay, we have shown that the same happen when we express Fabry alpha-Gal A mutant version in the brain, exactly the same. We express a misfolded enzyme, we create UPR in the brain, in the dopaminergic cells, and that causes the manifestation of Parkinsonian signs. And you know, we wondered what happens if we express Niemann-Pick disease AB related mutant forms. So I don't think I have to give huge introduction to this audience. Niemann-Pick is associated with mutation in the SMPD1 gene. Sphingomyelin is not correctly degraded to ceramide. So we have accumulation of the substrate, and we have NPDA and NPDB with an intermediate form. NPDA is a progressive neurological disorder. Interestingly, it's also prevalent among, like Gaucher disease, among Ashkenazi Jews with some pathogenic variants in Ashkenazi Jewish population, and I'll be talking about the L304P variants. In some cases, it will say 302, and that's because of different nomenclature due to two amino acid difference in the signal peptide. Never mind. And the second type is NPD, which is mostly non-neuropathic disorder characterized by progressive visceral organ abnormalities, hepatosplenomegaly, pulmonary insufficiency, cardiovascular disease, and survival into adulthood. So, you know, we went into the literature, see what's known, and we were amazed to see a paper which stated that Western blotting and fluorescent microscopy showed that the mutant ASM polypeptides were expressed at normal levels and trafficked to lysosomes. And I've just told you that misfolded proteins undergo ERAD, which lowers their steady state level in the cell. So this was kind of interesting and puzzling. And then we found also another paper that claimed that here you see 302, it's the same as 304. For 302 mutation in the lysosome enzyme SMPD1 is a risk factor for Parkinson's disease. So we have association with Parkinson's disease. We don't have any change in levels of mutant proteins in comparison to wild-type protein. What's going on here? And the third very interesting publication was from Alcalay et al. Showing that ASMs with the 302, which is a 304 mutation, failed to reach lysosomes. And it is ER localized. So we have something which is ER localized. It doesn't undergo ERAD, associated with Parkinson, a real puzzle. So we decided to express the mutant some mutant SMPD1 variants as transgenes in the flies. So what actually is being done is that normal cDNA or mutant cDNA, and you can see the 3 mutants that we chose, 294 is associated with type B, 304 and 498 are more prevalent among Ashkenazi Jews. This is the one was associated with Parkinson's disease. This one has never been associated with Parkinson's disease. So we expressed normal or these 3 mutants in animals as a transgene, which means we introduce 1 copy of a cDNA with these mutations in the fly. This doesn't cause anything in the fly. The gene is completely inactive. In order to get the gene active, and this is our gene, okay, which is coupled to an upstream activated sequences, which is a yeast promoter, which is completely inactive in Drosophila. So in order to make this gene active, we have to cross the flies with another fly that has an endogenous enhancer that activates the GAL4 protein. GAL4 is a transcription factor for the UAS. So once we have the new fly with these 2 genes, these genes makes GAL4. GAL4 binds to the UAS promoter, activates our gene, and the gene is now active wherever this enhancer was active. In our case, in most experiments, we express the gene, of course, through this enhancer in dopaminergic cells. And we ask, what happens in these flies? Okay, so what we were looking for, of course, what we were looking for unfolded protein response for this stress, because we have already shown in GBA1 and in alpha-Gal A flies that this UPR causes the death of dopaminergic cells in the brain, motor disabilities, and shorter survival. And we asked, can we, I forgot to write here that of course we also, it's here, death of dopaminergic cells. So we asked, does it happen with the SMPD1 variants? Okay, so first of all we asked, do we have protein? You remember the first slide I've shown you showed that the levels of SMPD1 variants are more or less the same. If you just take these, if you look at the 2 bands together, yeah, there's no significant change between the levels of the wild type and the different mutants expressed now in Drosophila. Interestingly, the upper band is the ER band, and the lower band is the lysosomal band, okay? And so, as you can see, the L304 is more abundant in the ER in comparison to the other mutations. Anyhow, so we have more or less still of the same, more or less the same amount of protein in the cell, though we are talking about wild type versus 3 mutants, 2 are associated with severe Niemann-Pick A. Very interesting. So we decided to ask, okay, do we have in any case ERAD? We have to test it. So in order to see whether we have proteasomal inhibition, what you do, you add a proteasomal inhibitor, MG132, and it turns out you can feed the flies with everything, including 2, they don't care, and they eat it, and they are doing okay. And we asked, if we feed the flies with MG132, are we going to get to see stabilization of the mutant protein? And as you can see, we get nothing, which means there's no ERAD of the mutant SMPD1 variants. Okay, this is interesting. What about UPR parameters? I won't go through the different parameters because, as you can see, there's nothing to say, nothing. Of course, you see different genes, UPR-related genes that we tested, and mutant SMPD1 variants do not activate UPR in adult flies grown at 25 degrees. What about expression, what about death of dopaminergic cells? Nothing. There's no death of dopaminergic cells. No change in TH levels between the different flies. Grow at 25 degrees, 35 days. This is very long. So, wow, interesting. And then what about movement? And movement you can test by the ability to climb on a test tube, on the edge of the test tube. And as you can see, normal and the 3 mutants are doing exactly the same. Of course, flies perform less well with age. And if we looked at survival, again, nothing. So the mutant SMPD1 variants do not present change in motor disability, nor in survival compared to flies, expressing wild-type SMPD1 variants. So interesting. So we decided to test the same parameters also at 29 degrees, which is a non-stress in the flies. And sometimes with a little bit of stress, the flies do show some phenotypes. Unfortunately or fortunately, we didn't see any change. So mutant SMPD1 variants grown at 29 degrees didn't activate UPR, no change in TH levels, no change in climbing, no change in survival. So nothing with nothing, as we say. Mutant variants do not present change in motor ability nor in survival compared to flies expressing the wild type. So there was nothing. So we have mutants associated with severe disease, which do not, one of them is in the ER, but they do not undergo ERAD, do not activate UPR, and if they do not activate UPR, we cannot see anything in these flies. So what's going on? So it turned out that in the ER, there's not only one stress response mechanism, the one I was talking so many years about, which is the UPR. There's another mechanism which is called ER overload response. So if this is the A is the UPR mechanism where we have 3 receptors which undergo changes and then culminate in transcription of UPR-related genes. The ER overload mechanism is associated with calcium release from the ER due to the presence of accumulation of misfolded protein in the ER. There's calcium release from the ER, which causes activation of NF-kappa B, which is bound to I-kappa B, but then it becomes released. N-kappa B is released from the inactive complex, enters the nucleus, and turn on pro-inflammatory genes. Also not mentioned here, there's also ROS in the intermediate production. And so the question was, everything fit our model, because in this case, as you can see, there's no ERAD, no reduction in the steady state levels of the mutant proteins. So exactly what we saw. And the question was, of course, will we see inflammation in the flies? And the inflammation was unbelievable. While we didn't see anything else, inflammation was unbelievable, and it was, of course, age dependent. So if this is 15 days, 30 days, and 45 days, see what we have. We have 400-fold activation in the 304 mutant, with much less production of inflammatory genes in the other mutations. So there is activation of neuroinflammation in the mutant SMPD1 flies. And so what we think is that, that our results indicate ER localization of the 304 mutant SMPD1 variant, strongly implying that is misfolded. However, this misfolding does not activate UPR. It seems to activate the ER overload response. This misfolding and EOR activity, we think, explains the association of this mutation of the SMPD1 gene with Parkinson's disease, and we are in the midst of experiments to show calcium release from the ER and production of ROS in the flies. And so I would like to end with thanking my, these are my present students. I have a small lab right now, and Aparna did most of the work, present work, with the beginning by Hila, and thank you for your attention.
Okay, thank you very much for the very nice invitation to come here today. My name is Aitor Aguirre. I'm an associate professor of biomedical engineering at Michigan State University. Our lab is it's pioneering the introduction of synthetic heart models, organoids, to some extent, but you'll see during my talk today that we have even taken things farther beyond organoids, and we are starting to work with very sophisticated models. And sorry, these are my disclosures before I move forward. Yes. I don't have any disclosures today related to lysosomal disorders. I am involved with 2 companies, but they have no involvement with lysosomal disorders, so really nothing to disclose here. Why hard organoids? Why are we interested in introducing these models? We are interested in these models because they are human-centric. We want to move basic and translational research into these human-centric models so that we can rely on the fact that we are working in humans and not in animals. So we can bypass some of the limitations of animal models. And how do I? Oh, yeah. And so this is a very timely moment because as you have probably seen recently, the NIH has started to get interested in this non-animal models research, or NAMs as they are calling them. Sorry. And so this is a priority now, or it's becoming a priority. So let me tell you a little bit, how do we make these human models? I'm gonna be spending a lot of time trying to convince you that this is something that it's valuable for your labs. So over the last couple of decades, we have seen the introduction of pluripotent stem cell models. For those of you that don't know what pluripotent stem cells are, these are cells that are derived from either the embryo, the very early human embryo, and they are the cells that make our bodies, or they can also be derived from adult humans in the form of induced pluripotent stem cells. So basically, these are stem cells that are at the top of the stem cell hierarchy, and they can be coaxed, they can be induced to become any tissue in the body. And over the last, I would say, 10, 15 years, that's what we have been doing. We have been playing around with these cells, and we have been differentiating them into different tissues. We have been developing protocols to create different cell types with them. And that's a little bit what you can see in this summary here. We have been able to make liver cells, cardiac cells, brain cells. But one thing that, and they have a huge advantage in this area because this constitutes an unlimited source of human tissue-specific cells, right? But one of the limitations that we have been finding is that these in vitro models, these cellular in vitro models, they lack complexity. They lack the physiological complexity that animal models have, right? And so something that has been happening in the last, I would say, 5 to 10 years is that we have started to try to achieve more complex in vitro systems that are more like organs. And that's what we call organoids. Organoids literally means like an organ, right? And so a new wave of research is aimed at what we call self-organization, which is basically taking advantage of the fact that stem cells know how to organize themselves. They know how to make these organs already. They have a blueprint inside of them that tells them how to make these organs. And so if we can, let's say, leverage, utilize these innate blueprints that the cells have, then we can make these organs in vitro with relative ease. And that relative obviously goes between quotes because it's actually quite complicated, but it does work pretty well. And so a strong influence here is going to be development, learning how these pathways work through development. And so by making these in vitro organs, then we can increase the physiological relevance of these human-centric models. Now, how is this important? How is this relevant to lysosomal disorders, which is what this conference is about? So this is important to lysosomal disorders because a lot of these lysosomal disorders have important cardiovascular complications, right? And many of the patients with these disorders will live shorter lives because of cardiovascular complications, because heart failure, because cardiomyopathies, cardiac fibrosis, arrhythmias, etc. And so I've put here a couple of them, but there's many more, right? And so these synthetic heart models can help us understand the physiological mechanisms and help us in therapeutic development for these lysosomal disorders. So how does my lab, which has been, we started doing this in the late 2010s, or like in 2019 or so is when we started introducing these models. How does my lab approach this problem? So what we do is we look at the heart and we reverse engineer how the human heart forms. And so these are a couple of things that we know about the human heart. It starts forming from a population of stem cells that is called the first heart field. I'm not going to go a lot into the developmental details. If someone wants to ask about this later, I'll be happy to explain. And then they organize, there's a second wave of cells that continues forming the heart. It's called the second heart field. And then eventually there's a third wave of cells that come, and it's called the proepicardial organ. And then there is external cells that come from the ectoderm, the cardiac neural crest cell, and these put the fine points, they finalize basically the structure of the heart. They don't come from the mesoderm, they come from the ectoderm, so basically the tissues that give rise to the brain. And that cardiac neural crest population will give rise to outflow tract and mesenchymal tissues in the heart, but also the part of the innervation of the heart. And finally, there's contribution from immune cells that are resident immune cells in the heart. These are called embryonic macrophages. Sometimes people call them primitive macrophages. And these will also help shape the conductance system in the heart and several of the fine details in the heart. And so what we have been doing in the lab is we have been recapitulating these different steps in addition. I'm going to give you examples of all of these in the next couple of slides. But what is very important to remember about this is that when we perform these experiments, we have been taking design considerations into our priorities, which are basically improve the reproducibility, improve the efficiency of these protocols, and improve scalability and throughput. And the reason for this is that if you are familiar with organoids to any extent, you might have heard complaints about how reproducible or how scalable these protocols are. And in the past, they have been not very scalable and not very reproducible. That is the truth. And so we are moving towards more standardization in this field. And I'm very glad to say that our method is very scalable, very reproducible. And I'm going to give you an example later on of one application that really needed those characteristics to be successful. Okay. So how do we perform this self-organization approach to build a human heart? So first of all, we want to create that first heart field and that second heart field. We can do that using, again, recapitulating development. So we can use chemical compounds and some growth factors, signals that develop the heart in early stages during embryonic development. And they will induce the formation of this layer of the heart. It's called the cardiogenic mesoderm. I'm not going to go a lot of detail, but this gives rise to the ventricle, the atrium, and parts of the outflow tract. This is basically the first stage of heart formation when you form the heart tube. And this is how we implemented this strategy. So basically, we take induced pluripotent stem cells, we seed them into 96 well plate, we assemble them into tiny little balls of cells that are kind of like little embryos. And then we implement these steps here with drugs, and this will assemble by itself and will give rise to these spheres of cardiomyocytes, as you can see here, that maturate and they develop striations, etc., like sarcomeres. And we know that this is very reproducible because we have made this across multiple different cell lines, whether it's embryonic stem cells or induced pluripotent stem cells. We can reproduce this. At this point, we probably have used more than 20 or 30 lines, and this is a very reproducible protocol. And that gives rise to that first scaffolding of the heart. But this is still not what I would call a heart organoid. This is just an assembly of cardiac cells. But if we go ahead and we introduce this third step that's called proepicardial organ induction, which is the farthest step in development, suddenly the complexity of these organoids skyrockets and just shoots up by a lot. This proepicardial organ, which is induced by a third activation step of Wnt, will give rise to the epicardium, the proepicardium, the vascularization, the valvular tissues, mesenchymal tissues, and also the conductance will promote the formation of conductance tissues in the organoids. And sorry, this thing is not working very well, yes, for the, perhaps it needs batteries. So when we do this induction, we get organoids that develop that myocardium that you saw before, but they also develop chambers inside. As you can see here, these chambers segregate and have these chambers segregate and have atrial and ventricular identities, and the external area of the organoid becomes an epicardium. So as you can see, this is basically mimicking the anatomical structure of the human heart. Here you can see electrical activity, and here you can see what a standard plate of organoids looks like with the little tiny organoids in each one of those wells. So this is very scalable. We can make thousands upon thousands of organoids, and we could make this from multiple patients if we need to. These organoids develop, as I said before, the first and second heart fields, and these first and second heart fields will develop atrioventricular specification, and this atrioventricular specification, this is two independent chambers. This is not interpeaks atrial and ventricular cells, but actually they segregate into both chambers. And obviously there's chambers inside. And this also develops cardiac fibroblasts, endothelial cells, and vascularization inside of the organoids, and also endocardial cells lining the inside of these chambers, which is the structure that we want to observe. So if you think about these organoids, they basically have all the key features, all the key structures of the human heart. Now, we have continued developing these protocols. Now we are looking at what I like to call heart organoids 2.0, because we know there are more refinements that we can implement here. And so what I have been showing now, it's more related to how the heart develops and the heart forms, right? But we can also introduce metabolic and maternal fetal environment signals around these organoids to promote further maturation and more physiological relevance, and that's what my student Brett Bolmer did in the past, and this is published. And so Brett designed what we call developmental maturation or developmental induction media, which is basically these developmental signals. He also designed more adult-like, He also designed more adult-like medium that we can use to maturate these organoids beyond embryonic and fetal stages into more adult-like stages. And this is an example of these organoids. So here you can see an organoid with ventricular and an atrial chamber here. In green, you can see the blood vessels. And you cannot see it here because it's not stained, but around the outside of this organoid, there is an epicardium. And this is an empty chamber. So all of this is a chamber, and all of this is a chamber. If you compare one of these organoids to human hearts, human fetal hearts, by transcriptomic analysis, they are almost completely identical. 96% of the cell transcriptome is identical. And the parts that are missing are mostly tissues that do not develop from the mesoderm, which is that cardiac neural crest population that I mentioned before, and those macrophages that I mentioned before. These developmental induction conditions that we introduce promote the progressive metabolic maturation of the organoids. And here you can see how organoids in control conditions versus maturation conditions. We have three different conditions here. stands for maturation media. This is a medium to bring the organoids to an adult-like state, while EMM1 and EMM2 are developmental induction media to recapitulate the steps of embryonic development that go from early stages, like about 2 months, into 6, 9 months old hearts. And you can see how these maturation media conditions promote oxidative phosphorylation and energy utilization that is more consistent with cardiac physiology, cardiac metabolism. Here you can see how those conditions induce the formation of atrial, nodal, and ventricular action potential in these organoids. You can see an organoid, the surface of an organoid beating there. And here you can see the expression of different molecules involved in electrophysiology channels and molecules involved in calcium handling and metabolic activity in the organoids. An interesting feature of these organoids is that they also recapitulate anterior-posterior patterning. So that is a feature that hasn't been observed before in vitro. What we observed in these organoids with our collaborators was that these organoids produce endogenous retinoic acid, and this is the molecule that actually patterns the heart tube to become atrial and ventricular. And we prove with our collaborators that this is true in vitro and that our EMM21 media conditions induce anterior-posterior patterning. And we also show that if we abolish that retinoic acid gradient, we can destroy the patterning of the organoids. And so what I'm trying to show you here is that we can make these very, very precise, very human, physiologically relevant heart models in vitro. But we can take this even farther away. Right? I'm going to show you how we can make this even more relevant. So we have been working also with heart assembloid models. So what are assembloids? Assembloids are, in the field, usually people refer to assembloids when they are combining different types of organoids. So this is a very popular approach in the brain. So using, for example, cortical and another area of the brain organoids, you can fuse them. In our case, what we are calling assembloids is these structures in which we actually need an external layer to integrate into the organoids to give rise to a more relevant organoid, right? So in this case, the cardiac neural crest cell, we create neurospheres, and we can culture these neurospheres with our heart organoids to induce, to mimic the integration of the human neural crest in the heart. You might be wondering why it's important to get this integration. This is important because the neural crest is involved in a lot of diseases that are related to congenital diseases, but also it uses the innervation of the heart, for example. And so we developed this protocol where we make these neurospheres, we make our heart organoids, and then we combine everything. And these are autologous cardiac neural crest cells. So these are derived, again, from the same cell line. So they can be made completely patient isogenic, right? Autologous, sorry. And so these neural crest containing organoids, as you can see here, these red balls, those are the neural crest cells. So when you culture them together, initially the neural crest cells are in a little ball, and then they will spread and they will start invading the organoid and forming nerves and other structures. Again, if we do single cell sequencing analysis and we compare to human hearts, we can see that our neural crest cells actually fall directly into the pool of neural crest cells that are migrating into the heart. So we know that our neural crest cells are very high quality and cardiac related. And this is a very busy slide. You can see that they develop nerve structures inside of the organoids, and these nerve structures are parasympathetic nerves. So this is the type of nerves that the neural crest actually makes in the heart. And this is important because these nerves are functional. And these nerves, as you can see here, we can put parasympathetic agonists and antagonists, and we can accelerate and slow down the beating of the organoids by triggering the nerves, not by actually modulating the cardiomyocytes, which is very interesting because these parasympathetic neurons are innervating the conductance nodes, they are innervating the cardiomyocytes. And so this establishes a good model for the study of arrhythmias in the heart. And this is an example. There you go. Here you can see those nerves inside of the organoid. So this is inside of the organoid. You can see them beating. Well, you can see the cardiomyocytes are beating. The nerves are just moving with the beat, right? So they are because of the deformation of the tissue around them. These are neurons that are labeled with M-cherry, so they are genetically labeled. The other generation, new generation of human heart assembloids that we are making is human heart macrophage assembloids or immunocompetent heart organoids. And this has been spearheaded by one of my students. This was published, actually it says in press here, but this was published 2 weeks ago in Cell Stem Cell. And so what Colin did is he developed a protocol to make embryonic primitive macrophages and physiologically and developmentally, in a physiologically and developmentally relevant fashion, include them, integrate them into the organoids. And this is a little bit the strategy that he followed. So he studied mouse development and human development and saw at what stages during development these macrophages move into the heart, what type of receptors they express. And so with that, he could find the optimal window for integration. I don't have time to explain to you all the details of the integration protocol, but believe me, it works very efficiently. Here you can see heart organoids with different concentrations of macrophages added to them, and the macrophages are the green cells here. And so we found out that about 20,000 macrophages is the optimal concentration in our organoids, because that represents about 2% of the total mass of the organoids, which is the relevant amount of macrophages found during development. So we want to make sure that we don't put too many or too few. And here you can see single cell plot maps that show that these macrophages express embryonic markers. The most interesting marker here to see is CCR2. That you don't want to see, because CCR2 is a marker of M1 and M2 type macrophages. So those are not cardiac tissue resident macrophages. So you don't want to see CCR2. And that's precisely what we see here. And then with these macrophages, with these organoids, we can do very sophisticated communication and physiological, crosstalk analysis. And here, this is an example of that. This is a cell communication plot. So here you can see the different cell populations in the organoids: ventricular cardiomyocytes, valvular cells, atrial cardiomyocytes, conductant cells. This includes the sinoatrial node and atrioventricular node, cardiac fibroblasts, epicardial cells, endothelial cells, and the macrophages. And you can see how the macrophages talk to these other cells and how these other cells talk to the macrophages. So this has very strong potential for mechanistic discovery. This is an example of a macrophage, again, M-cherry genetically labeled, inserted into the cardiomyocytes in situ and beat in unison with the cardiomyocytes. This is actually a feature that has been observed in mice. And this is the first time this has been shown directly in live human tissue. These cardiomyocytes inserted and passing electrical signals through them in the heart. So why are we making all these models? We are making these models because we obviously want to look at translational applications, right? And so in the context of this talk, I would like to ask you, what would you do if you had access to hearts of patients with, for example, Fabry disease, right? If you had access to as many hearts with Fabry disease from multiple patients, let's say dozens, hundreds of patients, what could you do with them? I think, I'm not going to answer that question. I think you guys have to answer that question, right? I have my own ideas, but I think it would be very interesting, right? Because you can use that for therapeutic development, you can use that for mechanistic discovery, you can do a lot of things, preclinical development. And so in our lab, we were very interested in a condition that's called pregestational diabetes, which is when the pregnant mother, is diabetic during the first trimester of pregnancy. This is very bad. This is very deleterious for the embryo, because the embryo does not have glucose control. The pancreas has not developed yet. And so at these early stages, diabetes on the mother has a very negative effect on the embryo, and it increases the risk of congenital heart defects from 1% in the common population to more than 10%. And so that is a very high risk of congenital heart disease. And so what we could find in our model is that we can actually recapitulate all the features of pregestational diabetes-induced diabetic cardiomyopathy, embryonic cardiomyopathy, very well. And these features, they have been observed in mice, right? So we can look at mice models, and then we can recapitulate that. So we see, for example, cardiac hypertrophy. We see mitochondrial stress. We also see endoplasmic reticulum stress. And I want to highlight how one example of how this model can help us discover new things. So we did single-cell sequencing in these organoids. These are normal organoids, and these are organoids subjected to pregestational diabetic conditions. And we found out, we confirmed what people already know: cardiomyocytes are very affected in this setting. But we also found something very interesting. We found that the epicardium in these organoids is dysfunctional in these diabetic organoids. It's actually very dysfunctional and doesn't process fats and fatty acid synthesis in a relevant way. Now, you might be wondering, why is this relevant? Well, this is relevant because this is something that you cannot find out in mice, because the epicardium in mice is very different from the epicardium in humans. And actually, mouse do not develop epicardial adipose tissue. So the epicardial tissue in mouse has a different function than it does in humans. And so this is a very human-centric phenotype. And there might be many of these phenotypes that we are missing in our animal models that we could discover in these human-centric models. And so I hope that I have convinced you that these synthetic heart models are powerful tools for disease mechanism studies in humans. We know that there is still a lot of refinement necessary for these models to reach full potential, but they are already very powerful tools for disease modeling and therapeutic discovery in vitro. And with that, I will go over my acknowledgements, my team, some of my team. There's some people missing there, my collaborators, especially my clinician collaborators. I haven't talked a lot about clinical collaborations, but we have a lot. This is our institute at Michigan State University, and this is my founding support, mostly from the NIH, the American Heart Association, the NSF, the health system in Michigan, Corewell Health, the Alternatives for Research and Development Foundation, and the Saving Tiny Hearts Foundation. And yeah, with that, thank you.
Thank you. I have the pleasure to introduce Dr. Ying Sun. She is a professor and principal investigator at the Cincinnati Children's Hospital Medical Center and focuses research on LSDs, glycosphingolipid metabolism, and neurodegenerative diseases. Her talk today will be the generation of a brain organoid platform to investigate neuropathic Gaucher disease and therapeutic strategies. Dr. Sun.
Thank you. Thanks, Alexander, gave me the opportunity to present our work, and also appreciate the previous talk already discussed the importance of modeling disease use organoids. My talk will be focused on the neuropathic Gaucher disease, and we generate the brain organoids and to study the disease mechanism and drug testing. This is my disclosure and the learning objectives. So neuropathic Gaucher disease is caused by the mutation on the GBA1 gene, which leads to the defective acid beta-glucosidase function. And in brief, we just call this a GCase, and which also causes the substrate accumulation, glucosylceramide and glucosylsphingosine. And the Gaucher disease is classified for 3 forms. GD1 primarily is visceral disease, and GD2 and GD3 are classified as a neuropathic Gaucher disease. And the GD2 is more severe than the GD3. So our modeling is focused on the GD2. Here we showed some images from our study and also from Dr. Stransky's study showing in the patient brain. And you can see the top H&E stained image showing the neuron has a storage materials, and the bottom image showing the region in the middle brain of the patient, GD2 patient, and has a perivascular involvement. Not just the degenerated neurons, also has those microglia cell cluster. And we'll also analyze the substrate level in the brain at a different region, and you can see as a point there, the basal ganglia, which is within the midbrain region and has a highest level of glucosylceramide accumulation. These findings suggest the midbrain is a pronounced region in the neuropathic Gaucher disease. That's the focus in our disease modeling. And then from the previous studies, our lab and other researchers also demonstrated in the neuropathic Gaucher disease, due to the substrate accumulation, there's a disturbed cellular function, include autophagy, lysosomal, mTOR pathway, and also ER stress and mitochondrial dysfunction. most of these studies was done using the cell model and the animal model. Cell model currently used if for the human study is from the patient or healthy individual primary fibroblast cells. Some lab also develop neuronal cell lines and by manipulate either like IC40 into induce transformed cell lines. And then recent years, very popular cell study is using the induced pluripotent stem cell derived cells. In terms of the animal model, my lab and as well as other labs has developed many mouse models, include a knock-in model, knock-in mutation model, knockout, transgenic model, and also chemical-induced model using the GCase inhibitor. However, those models do have a limitation to study the human Gaucher disease, especially the neuronopathic Gaucher disease. Like human cells lacks complexity and functional capacity, and the animal, especially the mouse brain, do not identical to the human brain. And currently, the human sample mostly from the postmortem brain tissues. So these definitely is, have challenges to study the neuronopathic Gaucher disease. Recently, the organoid technology has very rapidly developed, and so for modeling the neuronopathic Gaucher disease, like we can use 3D brain organoids. So 3D brain organoids is a miniature and specialized neurons can grow in the dish, and is can be used can be derived from human cells. Especially, we can derive from human patient cells. And these brain organoids consist of multiple neural cells and organized in a relatively similar brain structure. This technology has applied to several neurodegenerative diseases, include Parkinson's disease, Alzheimer's disease, and also lysosomal disease, include Gaucher, neuronopathic Gaucher disease. So in our lab, we developed this NGD patient iPSC derived midbrain-like organoids. I like call this like organoids because this is still not represent true midbrain structure. So in our study, we include those cells, healthy iPSCs, which we call the Y-type cells, and we have two GD2 patient iPSC lines. One is generated in my lab with when Dr. Grabatsky still there, is GD2-12660. Another line, GD2-10257, is provided by Dr. Feldman, and I think the original is from Dr. Stransky patient cells. And also, both of these lines carry the L44P mutation, which is a common mutation in the Gaucher disease, and combined with other different mutations. We also generate isogenic control lines. iPSC use GD2-1260. So we use this model to, we use these iPSC cells derived to MLOs and study the disease phenotype. And also we try to test several ongoing therapeutic approach for the neuronopathic Gaucher disease. This is a diagram using healthy iPSC cells to develop midbrain-like organoids. This work mainly done by Dr. Ying, very talented postdoc fellow. Unfortunately, he moved to the industry. Yeah, so in this environment, just the scientists move away from the research, very unfortunately. But using this protocol, mainly we follow the published protocols with minor modification, especially on the percentage of microgel used to generate successfully generate MLOs. So when we analyze these MLOs on the 8 weeks of differentiation, you can see in our model there's neuroprogenitor cells. We also have neurons positive for neuron can't really see that. I think this, okay, TOJ1. And these organoids also contain the astrocyte, positive for the GFAP. We also, because this is the midbrain organoids, we also tested if they contain the dopaminergic neuron, which is a positive for FOXA2 and TH markers. And to confirm this is midbrain-like organoids were compared to cerebral organoids, which is positive for FOXG1. And you can see the MLO does not contain this signal. So with this protocol, we had generated the NGD MLOs using GD2, using one of the GD2 cell lines. And then we compared the organoid growing size at a different differentiation stage. And you can see they don't show the difference compared to the healthy MLOs. So indicate that the mutation did not affect the organoid growth. And then we analyzed the GCase protein and activity. They do show the decrease. And also they do show the substrate accumulation for glucosylceramide, glucosylsphingosine. And we also analyzed the substrate species because in the brain, normally the 18 chain lines is predominant in the brain. And again, in these organoids, and the 18 also is predominant, also accumulate. And then we also analyzed the gene expression in these NGD MLOs compared to the wild type. And you can see there are several pathways show the differential expression, include lysosomal, autophagosome, and neuronal function, etc. Among these, we found quite interesting, like FOXP1 and FOXG1. Those is midbrain patterning or forebrain patterning marker genes show the differential expression in the NGD MLOs. So this suggests there's abnormal expression of a fit determining regulators in the NGD model. Again, this is immunofluorescence staining also support this finding. In the NGD MLOs, we also found some dopaminergic marker genes like TH, FOXG1 also show the reduced expression, and also some lysosomal marker genes, LAMP1 and cathepsin D also show the decreased expression. So then we interesting to ask the question whether dopaminergic neuron differentiation also changed. And indeed, when we analyze TH and FOXA2 markers, and those both markers show the decreased expression. These also confirmed by immunoblot to measure the protein level. Both markers show the reduced expression. And then whether those dopaminergic neuron function also affected, then we measure the dopamine level in the cultural medium of NGD MLOs, and indeed it reduced compared to the normal controls. So NGD patient iPSC-derived MLOs do present the key GD phenotypes, include the decreased GCase enzyme function and accumulation of substrate. And it also showed the altered transcriptome profile affect several cellular pathways. And interestingly, it's also present the impaired dopaminergic neuron differentiation. So I think this model should offer a patient-specific 3D neural models for studying the disease mechanism and also for testing the drugs for NGD. Currently, the new therapies in development for NGD include gene therapy, CNS enzyme therapy, small molecule therapies. So I just briefly talk about some of our testing result in the next few slides. So to test those therapeutic strategies, we include 4 approaches here. One is genome editing for gene correction using CRISPR-Cas9, and second is substrate reduction therapy using small molecule GZ452. This is analog of venglustat, which is the SRT for treatment of neuropathic Gaucher disease. Right now is in the clinical phase 3 testing. And an additional approach include CNS enzyme therapy. In this case, we use a substance DOPS liposome to carry the GCase for deliver into the cell or into the brain. And finally, the gene therapy. So among those gene correction, enzyme replacement therapy, and the gene therapy basically is to provide a normal enzyme to the cells. And in contrast, the small molecule substrate reduction therapy is to inhibit substrate production to reduce, to balance the substrate level in the patient cells. These slides showing the mutation correction use a CRISPR-Cas9, we correct the IO44P mutation to the Y type. So basically, the resulting IPIC lines is carry the isogenic GBE1 mutations. Then we also measure the MLO size to confirm this isogenic change. The mutation correction did not affect the organoid growth. So then we compare the GCase protein level, GCase activity level, compared to the NGD with isogenic compared to the NGD original. And you can see there's about close to 50% recovery of the enzyme. Then the substrate level showed a significant reduction, even close to correct to the Y-type level. Yeah. Sorry, I think there's one. Okay, yes, both in the medium and in the MLO cells. And then we also ask whether this the mutation correction can recover the dopaminergic neuron differentiation. From this result, you can see the TH and the FOXA2 marker are recovered to Y-type or close to the Y-type level. And also dopaminergic dopamine level also recovered, suggests there's a rescue of a dopaminergic neuron in these NGD MLOs. And then additional cellular marker we studied is a lysosome and the mTOR. For the lysosome, LAMP1 expression was recovered in this isogenic line. Cathepsin D did not show any change. And in the mTOR pathway, the 4E-BP1, either phosphorylated form and unphosphorylated form, all showed improvement in the isogenic lines. So these mutation correction do rescue the key NGD phenotypes and also improve several downstream effects. So I think this result also validate the NGD MLs as a preclinical disease model, and also support the genomic editing could be a therapeutic approach for NGD treatment. So then these slides are showing some of the testing of a different drug. This is a SRT treatment. And first, we want to select the safety dose. So we studied these dose-dependent change in the healthy MLOs, and we measure the size. If the size not change, that indicates the dose is safe. In this case, the 0.3 micromolar, which is 300 nanomolar, looks like showed a significant reduction of the substrate, but not affect the organoid growth. So this is the safety dose we use to test in the NGD MLOs. This drug treated for 28 weeks and showed a significant reduction of the substrate. And this treatment also rescue or improve the lysosome function and also autophagy function. Next testing is using substance DOPS nanovesicle deliver the GCase. Actually, this approach we have demonstrated in the NGD mouse models, and this is already published. This is using substance DOPs, which form the liposomal vesicle, and it can formulate with GCase. In our mouse study, we use is a Y-type GCase. However, the Y-type GCase has a very rapidly degradation in the plasma stream. So recently, in collaboration with the Spur company, they developed a variant of GCase, which has a much longer half-life compared to the Y-type GCase. So we purchased this enzyme from their company and on the MTA, and then we use these FGCase formulated with substance DOPs treated on our NGD MIOs. In this experiment, we include 2 NGD MIO models carried different mutation. As you can see here, the green fluorescence is showing the GCase signal, and so the treatment do show the in both patient MIOs, the GCase uptake into the organoids and also localized in the lysosome and the autophagy, indicate that they have a recovery of a lysosome and autophagy enzyme function there. We went ahead, measured the GCase activity and the GCase protein. They all show the recovery close to the Y-type level, and the substrate also reduced in these treated NGD MIOs. Finally, we also tested the AAV approach, and the AAV was injected into the MIOs, and then after 3 weeks treatment, and we collect the MO, analyze the GCase expression in the cells. As you can see here, GCase is uptake into the neuron, in the dopaminergic neuron, and also in the astrocyte. Again, the GCase activity was recovered to about 50% of Y-type level, and the substrate also significantly reduced close to the Y-type level. So in summary, I think our study demonstrated the feasibility of growing NGD MLs to model the disease, and these NGD MLs do present NGD, the GD phenotype, include GCase deficiency, substrate accumulation, and several abnormal cellular functions. We also found that NGD MLs show the abnormal differentiation of DA neurons, and this was rescued in the isogenic line. And I think by test, this model will provide a platform for drug testing, which I have demonstrated 4 approaches. We can demonstrate there's a response of this model to those therapeutic approaches. And in the future direction, because these models still have a limited cell containing there, so we are developing advanced model by integrate the microglia cell into our MLO models. And also in the future, I also like to incorporate the endothelial cell into this organoid, so more closely mimic the true brain. Well, in perspective, I think these NGD MLs enable individualized precision model and also to support the precision medicine. And brain organoids has the potential to improve the preclinical testing and to reduce the reliance on the animal model use. Finally, I'd like to just thank the people contribute to this work. Benjamin Liu and Vinay Fanny, who contributed on biochemical analysis, and Dr. Yi Lin is a major contributor to generate these organoids. And my collaborators in the different departments in our hospital, and also Dr. Feldman, Dr. Sadrensky, by providing the cells. And my funding support is from NIH, and also from the several foundations. Thank you.
Thank you all for your presentations. This session is now open for questions.
Hello. They were fantastic, the organoid ones I'm very interested in. Dr. Aguirre first, and maybe I'll give my question for Dr. Sun second. What's driving the development of the mouse heart, 5 times quicker than the human one? Have you looked into that? And can you, and sort of as a corollary to that, can you speed up the development in the organoids to show the sort of late Fabry hypertrophy phenotype?
So regarding the mouse heart, I really don't know the specifics. I know that the internal clocks in the cells during development are different for different species. So the human heart simply develops at a slower rate. I think this is evolutionarily coded into the cells. And there's been very elegant studies comparing mice, humans, pigs, and different species. And this is one of the reasons why interspecies chimerism doesn't work very well. So if you take, human cells and you put them in a pig heart and you try them to contribute so that the pig heart has human cells in it for transplants, this is interspecies chimerism is very interesting for those translational applications, right? They don't really contribute too much to the pig. There's like only trace amounts. It's because they cannot follow up with other species rates of development. But I don't know the specifics about the mechanisms inside. But I can give you a very good answer to your second question. Our work branches in 2 directions, right? So one of the directions is congenital disorders and pediatric disorders, because to get to the adult forms of the organ, we necessarily have to go through the younger versions, right? But despite being able to, model those developmental aspects, we can also push the accelerator and we can force the cells to kind of like move a little bit forward. And that is doable, and we have done it in very recently in that cell stem cell paper I mentioned. We actually model atrial fibrillation, adult atrial fibrillation in the organoids after a 60 days maturation protocol that actually produces very adult-like features. It's not perfect, right? It's not adult like my heart or your heart, which has been in my body for 45 years beating, right?
Mine a bit longer.
But it's a model, right? So I always say that regarding models, you need to ask the right question to the model. Is the model able to answer your question? If it is able to answer your question, then it is a good model. So the question that we were asking to that model, which was atrial fibrillation, it was able to answer it very well. So in the case of Fabry disease, I think we would probably be able to capture significant aspects of the phenotype, right? But we haven't done it yet. Perhaps someone here wants to collaborate with us and explore it. I would be very interested in that because I think it's a fascinating area, especially in the case of late onset Fabry disease, which is very difficult to model in animals, I know. And I think that would be an interesting area to explore with the organoids.
And maybe something we could talk about afterwards is just something you said triggered the thought. There's a lot of diseases such as the ones that we're developing with Azopharos, which is with the early, late and adult onset of the same disease with similar mutations. So that's a challenge for us. But Dr. Sun, if I can ask, do your organoids organize themselves into the same layer structure that you see in normal brains?
No, not there yet.
Not at all. Okay. Thank you.
It's kind of like a piece of tissue, very, very small, very tiny. So, and also because we try to model GD2 and to mimic the early development, actually our organoid is about 8 weeks to 15 weeks, which is about the embryonic stage 9 to 10 weeks. So it's still a very early development stage.
that's on or not? Oh, it's on. Okay, thank you very much. So, two questions to each speaker. First of all, this organoid model can really, replace a preclinical trial or not, mouse or rodent, whatever level, which is skipping some step. That's quite important to go to FDA for sure. Second question is that, let's say, you can make the model, normally it's fine. Our concern is that always systemic disease, whatever LSD in general. So can really specific organ can go to overall, systemic disease or not? That's also quite important to know that. One more question that, let's say heart valve. If you have a normal heart valve, you can make it. Can we transplant or not? When we can do that? That's more specific questions of transplantation. That could be quite important. Many LSD have a heart disease, I know that. Then if we can take IPS from patients, then if we can transplant the specific tissues, that would be the more direct impact the patient, I guess. Thank you.
There were a lot of questions there. So I'm going to start with the last one because it's the one that comes to my mind right now. No, you cannot transplant this. Obviously, this is not ready for transplantation. It might be ready for transplantation one day, right? I think the hardest part, in the specific case of the heart, I'm going to speak for myself. The hardest part of making a heart is making those first 3-month structures. We are already 90% there. But once you have the template, once you have the blueprint of a heart with the different structures and the different organizations, then it's just maintaining it and letting it grow bigger, 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 right? The problem is that all of those technologies do not exist. We need machines, we need machinery, we need sensors, we need instrumentation that can maintain an organ that is going to grow several fold times in size in a very short period of time, right? So you have, at those early stages, our organoids are like 2 millimeters in size. And if you were trying to model a human heart, in 3 months, it's going to be the size of my fist, right? Well, perhaps a little bit smaller, but it's going to increase in size very fast. So you will need instrumentation to maintain that extracorporeally. That does not exist. So that requires engineers, requires experts in many other fields. So it is a challenge. But I think it's going to be possible one day, perhaps decades from now, right? What I can tell you right now is that our organoids, we can put them in animals, right? And so the animal acts as a bioreactor that maintains the organoid, and we have done that in mice, so we can implant them in mice. And the moment you implant them in mice, our organoids are anastomosed to the vasculature of the mouse, and in 2 weeks, they grow in size 5 to 10 times. Like they increase in size almost exponentially. They get very big. And you can actually MRI the mouse, and you can see the mouse heart, and you can see the human heart implanted too. And you can see the chambers, and you can see the beating of the heart. You can see the blood flow through it. So they have very strong, integrative capacities in that system. Is that ready for humans? No, it's absolutely not ready for humans, but perhaps one day. There were 2 other questions there. Sorry, I don't remember right now.
Can we skip the preclinical trials?
Oh, preclinical trials, yes. These are really good tools for preclinical trials right now. A lot of industry interest in these tools right now. I am co-founder of a company that is actually offering these tools for, big pharma like Pfizer, Johnson & Johnson, etc. So they can do preclinical trials in organoids. This is very interesting because it fills a gap in the preclinical pipeline of many of these companies. Because many times they go from highly artificial in vitro models, like, cancer cells that are expressing a receptor where they do a screening, and then it jumps directly into animals, right? And so and then they test it in animals, but, then they test it in primates, perhaps. And this is very expensive, and there's a very big gap there where they are taking kind of like a leap of faith. But if you had an organoid platform there for that preclinical development, that is a human-centric platform, you probably could identify a lot of drugs that should be removed from the pipeline, either because they are cardiotoxic or perhaps because they are not efficient if you are doing therapeutic efficacy. So In preclinical, I think it will be really, really very interesting. I think perhaps an even more interesting question is in clinical trials, like clinical trial on a dish. It's a concept that has been floated around by Joe Wu and other researchers in this field for years now. With patient-specific organoids, right? Like you could select a sufficiently large sample of the population that is relevant to the population makeup of the US, for example. You can select thousands of lines, and you could perform a clinical trial on a dish on specific drugs before they move into the market. And with AI tools now, like integration of the data with AI, et cetera, if you use a sufficiently large cohort, I think potentially one day we could be able to bypass a clinical trial. We could get sufficient, precise information that we don't need to do a clinical trial. That's obviously not going to happen now, but I think perhaps in the future. There was another question. I don't remember what it was.
I'd like to add on that because actually I'm the mouse model person. And I can see the gap there. As people working in the Gaucher disease field, you will know, very unfortunately, patient mutation, when you knock into the mouse, they don't generate the behavior or the phenotype like in the patient. So I think this organoid model will fill in this gap so we can have patient-specific mutation in these three-dimensional models. Although this, I also recognize this has limitations because this is not a systematic, all the organs in there, right? But if individual, different organ like heart, here, kidney or lung, liver, all have the organoid, then you can use these to screening the drugs to target the different tissues. So this definitely will provide important preclinical data, reduce the use of the mouse models.
So I remember now your first question. It was about the multi-organ, right? Like the physiology, like the organs that are missing. It's a great point. You obviously cannot model what's not there, right? So if there is a communication, that comes, for example, from the liver is dysfunctional, it's producing something that is affecting the heart. You cannot see that in an organoid model that it uses only one type of organ, right? Like the heart. But I'm going to turn your question around. And I'm going to say that can also be an advantage because this allows you to dissect the disease, right? So if you are looking at an animal model and it's a multi-system disease that is affecting multiple systems at the same time, and you're looking at a specific organ, it can be difficult to determine what other system it's fitting into that disease loop, right? Like there's compensation sometimes. In an organoid system, like for example with our organoid, you can turn this into a strength because you might be able to model the contribution of that other organ through, introducing something in the system, right? Like for example, is the liver and the liver is producing some kind of molecule that is toxic for the heart. You can add that molecule. You don't need to add a whole liver to see that interaction. So it can allow you to dissect the disease progression. So it can be a weakness, but it can also be a strength. And finally, we are getting closer also to multi-organ organoids systems in vitro, right? Like some people have already the Gordana, what's her name? Sorry, I forget her name. It's a very complicated name. It's a Serbian name. There's a group in university. It's Columbia, I think, University. Sorry, I cannot tell you the name of the PI, but I can find it if you really want to know. They have already published like multi-organ chip systems for multiple organoids, and so I think that's going to happen in the near future.
Hello, my question is to Mia Horowitz. So there's plenty of literature about unfolded protein response, UPR, but that second mechanism of air stress overload, is it a newly discovered mechanism or is it something known for a long time and just not as well illustrated in the literature? And second question, does it manifest itself in Gaucher disease?
Again?
Does it manifest in Gaucher disease?
Okay, so first of all, it does manifest in Gaucher disease. Mutations we have tested in Gaucher disease, there are more than 1,000, we tested few, and they all follow the UPR rule. And we were searching the literature because nothing matched UPR in the case of SMPD1, and then we came across this mechanism that was discovered or was published, I think like 20 or 30 years ago for the alpha-1 antitrypsin deficiency, and there they found several mutations. Some followed the UPR rule, and others, like if I remember, it's a Z mutant, followed the ER overload mechanism. So there were several groups that published it, and somehow this mechanism was abandoned. So we couldn't really find any literature about it in the last 10 years. She's talking to herself now.
There is something called ER preemptive control, and I think these two mechanisms, I think either they're the same, with semantics, or they do overlap.
Yeah.
So basically, actually, we were looking for the reason why 84GG would bring without any protein product, an ER overload or similar, why it's more represented in GDPD. Is that right? So one plausible mechanism was ER preemptive control, and this has been actually published. There's a PI at the NIH, I don't know if it's still there, but he did extensive research on the on Krustal-Jakob protein and similar. So actually, if you have a protein that is prone to misfolding, like synuclein, and having another one at the same time would enhance that mechanism, and then it overwhelms the system. It's in a nutshell. I have one question for Mia. So obviously, sphingomyelinase is a very bioreactive molecule, and it ends up inflammation as in general terms. But inflammatory process is responsible from the secondary glycosphingolipid actually production. No, you don't agree? I mean,
yeah, completely.
Yeah. Okay, so there are multiple companies that are coming from the haywire, proposing, GSL reduction as a treatment of GDPD, including enzyme therapy, so on and so forth. So did you look in your systems about the GSLs secondary deposited to the inflammatory component only, not from the primary, because when you treat the primary issue, yes, you can treat the disorder, but the disorder still moves on, like in GD2, is that right? We can treat those kids, but it becomes a super inflammatory disorder, and then it causes, all these fibrosis, scarring, and death at the end if there's a neuronal. So did you look at, what is the role of secondary GSLs when we control the disease, but still there is inflammation going on?
Yeah, don't forget, in our model, we don't have anything beside overexpression of a mutant protein in any tissue of the fly that we choose. In our case, it's the dopaminergic cells. So there's no GS cells, and there's nothing playing any role there. That's the... What's the point in measuring it? Nothing. No, because we don't change it. The normal enzymes are there. The flies are completely normal. These are normal flies. They have everything, all the normal enzymes they express all the normal enzymes. We did check it in case of the Gaucher models. So everything is there. So it's only expression of a mutant protein. And the question is, what does this expression cause in the specific cells it is expressed in, or maybe, in other cells? But there's nothing to do with any substrate accumulation, because it's a completely normal fly. This is like you take cells like HeLa cells, 293 cells, and you overexpress in them your gene. You don't change anything in these cells beside overexpressing a specific gene and looking what happens. in the cell. It's exactly the same.
Any more questions?
Well,
Okay,
one
one.
quick question. For me, just following up, you said that you have reactive oxygen species as well. Did you measure in the flies? And if that's real, is there a connection with mitochondrial stress or between the ER? And you said that the calcium is low. Eventually, the depletion is also in mitochondria that eventually causes this.
It is being tested. Yes. We are testing it right now. So we have to make the specific process in order to get the right flies, and then we are going to check the reactive oxygen species and intermediates. Okay, so we don't have final results yet. And mitochondria, of course, can be involved.
Then another question for Aguirre with regards to the organoids. So Gordana Novakovic is at Columbia. So she is.
That's what I was trying to say. Sorry, I couldn't.
So the main challenge, I think, when she connects the different kind of environments or wells that contains different organoids, and then connecting is to fine-tune the parameters that one organoid is impacting the other. So like brain, heart, kidney, liver, spleen. But she has been successful looking at toxicology, looking at screening compounds that eventually have teratogenicity as well as different impacts in hepatotoxicity, nephrotoxicity, and so on.
Yeah, we have done also a lot of pharmacology studies. I didn't present that, but we have been very successful at modeling cardiotoxicity by different types of drugs. And also we have been very successful at doing cardiac safety screenings, like compounds that are suspected to have congenital heart defects, for example, right? Like the drugs that women take that are suspected to induce defects in the fetuses. We have been able to recapitulate that in the organoids too, at very relevant, physiological parameters, basically. I don't want to go a lot into the details, but we can talk about more of that later if you are interested in that area. But yeah, I agree. I mean, there's a lot of challenges, and having multiple organs in the same chip has its own challenges, right? That need to be overcome. Yeah.
Hi, thank you for the presentation. I have two questions for organoids. So I have two questions for organoids. So first one is related to establishment of these organoids. So you said that you combine different type of cells to establish these organoids, and each cells went through different process, and in the end they were combined together. But these cells are coming from different culture conditions. So first question is, how did you decide the number of cells to combine each other? Is there any rationale behind that for different type of diseases, for instance, skeletal disease related or muscle? And second question is, how was these cells survival in the same culture conditions, since they were prepared in different cultures, and when they come together in the same media or culture related to organoids, did they survive, or how was their physiologic state? Thank you.
It is for me, that question?
Yes.
Oh. Yeah, so we don't actually culture multiple cell types independently and put them together. We culture one single cell type, that is pluripotent stem cells. And the different cell types spontaneously arise inside of the organoid. So they follow nature. We actually do not try to babysit the cells. What we do is you have the organoid, and the heart in there develops kind of like it would in an embryo. We know, with a lot of, take that within quotes, right? There's a lot of things that are not like an embryo, but it follows the developmental process. We don't go and make endothelial cells and cardiomyocytes and atrial node and ventricle. We don't do that. We just give the main cues that make the heart, and then the cells start branching out and taking differentiation pathways that are relevant to heart formation, and they establish themselves. And so they self-regulate. So the cardiomyocytes and the fibroblasts and the endothelial cells, they talk to each other inside of that organoid, and they regulate in a way that actually diminishes a lot the effort that we have to make it into a relevant heart. If we try to make that by taking all of those different cell types and put them together, I can guarantee you it wouldn't work, because people have done it a lot and it doesn't work. The fibroblasts tend to proliferate much faster than the cardiomyocytes. They take over the culture, the different culture media conditions, as you mentioned. All of those are problems. In our case, that doesn't exist. The cells actually know how to regulate themselves. They know the proportions. They know how to signal to each other. So it's basically, it's a community of cells. And so by following that developmental self-organization approach, it requires much less effort to make these organoids than it takes to make other types of 3D culture assemblies where you actually have to babysit the cells, right? So, for example, in our case, we can introduce cardiac tissue resident macrophages, neural crest cells, endothelial cells, etc. in those organoids. We don't need to provide any external growth factors to maintain those cells. The organoid itself produces a microenvironment that supports those cell types inside. So it's a very low effort type of approach, which is one of the advantages of it.
Thank you.